Hello everyone and welcome back to the session on glycosides. So far we have learned different type of glycosides, anthraquinone glycosides, we've learned different compounds. Now we are moving on to a set of very unique compounds, and those are cyanogenetic, isothiocyanate and phenolic glycosides. To start with the first category, that is cyanogenetic glycosides, these are glycosides containing a cyano group, that is a cyanogenetic or cyanophoric. Basically these groups have what is called as a cyano linkage. Have you heard of this gas or a liquid called as hydrogen cyanide? Now hydrogen cyanide is one of a very poisonous gas which acts as a contact poison, and for humans and for animals, if you carefully observe the effects of hydrogen cyanide, they in humans somewhere it inhibits the ability to carry oxygen smoothly when this gas is inhaled, and in animals they prove to be really lethal, often interfering with the electron transport chain. So cyanogenetic glycosides or cyanophoric glycosides are compounds, or you can say a plant's way of hiding this beautiful biological weapon called as hydrogen cyanide. Now this biological weapon hydrogen cyanide is hidden in a way that it is kept in a form which is conjugated with sugars. Now plants try to keep it. Now if the plant will try to save the hydrogen cyanide in itself, it will be toxic to the electron transport chain, or it will be toxic to the plant itself per se. So as a result, what happens, the plant wants to kill the animal consuming it, but it doesn't want to harm itself. So what it does is it kind of converts this hydrocyanic acid into set of compounds by adding sugars to it. So cyanogenetic glycosides are characterized as cyanohydrins, or also referred to as alpha hydroxynitrile derivatives, which are stabilized in the plants by glucosylation, wherein a sugar or disaccharide has been added to it. So you will see numerous such examples such as amygdalin, dhurin, linamarin and prunasin. Now what is the common feature of this is, suppose if you focus on this particular carbon, you will see bearing linamarin, all three of them have a hydrogen except here where there is a CH3. Then you have a cyano linkages, then you have a glucosidyl linkages, and in this fourth case you might have a phenyl ring. Now this phenyl ring again in linamarin is replaced by a methyl moiety. So little linamarin is slightly different, but you can generalize the other three. So now what is going to happen is, whenever an animal consumes this plant, now the plant wants to release its toxicity. So if you remember, plants have an enzyme called as glucosidases. So this glucosidases will gradually break down the linkages, from here, and as a result convert it into a hydrolyzed product. Now this hydrolyzed product later on will form a ketone and release HCN. This HCN which is released will then prove lethal to the animals. Now let us see this. So take an example where you have your linamarin, the simplest one. I am just drawing CH3 for your convenience. Now first thing what will happen is a glucosidase will enter in, and this glucosidase kind of acts here, releases your sugar, and what you get in the process are your derivatives. Now what derivatives you will get in process is first will be a hydroxy, little unstable, but yes, a hydroxy, what you call it as a hydroxynitrile derivative, and then this hydroxynitrile derivative will slowly convert it into aldehyde. Now as a result, what you get here is you get a ketone, and plus you will get HCN. This HCN gas or this HCN which is released is going to be proving lethal to your animal which is consuming it. So it's a way of plant's defense system. Now interestingly, the cyanophoric glycosides as well as the enzymes are spaced, that is in plants they are kept away from each other in slightly different cell organelles, but at the time of consumption, when the animal is consuming it, in the process of chewing, these organelles are ruptured, so glycosidase comes in contact with the cyanophoric glycoside. It has no option, but it is definitely going to lyse the sugar, and as a result your hydrocyanic acid is released. Once hydrocyanic acid is released, it causes all the toxic effects. Now one good example of a hydrocyanic acid releasing compound, or one such example of a cyanophoric glycoside, is your bitter almond. Now we have tasted sweet almonds, so it's a very close plant with a slightly different variety. So it's a Amygdala amara, or it consists of dry pride seeds which appear almost similar to your almonds, but they taste a little bitter, and this bitterness is attributed to the compounds present in it. So this is also Prunus amygdalis. When you are talking about the sweet almonds that we consume, it is variety dulcis, but when you are consuming a bitter almond, it is Prunus amygdalis, it is a variety amara, which is different. Both of them belong to the Rosaceae family and bear beautiful flowers. Now this bitter almonds have been cultivated throughout. You will see them in a slight temperate regions across wherever there is a little colder climate, Iran, Asia, you will find it in Sicily, Portugal, Morocco, and even in South France. Now this cannot be consumed as they are bitter, they are not clearly edible because they are toxic. Over a period of time, if you eat more of this hydrocyanic acid initially causes sedation and may prove lethal in humans also. But what you can prepare out of that are two things, that's your oil and flavor. Now chemically, if you see your almond, it contains, or chemically if you see your bitter almond, it contains a cyanophoric glycoside called as amygdalin. Okay. Now this amygdalin has two sugars attached to it, a phenyl ring and a hydrogen, like we saw generically. Now what is going to happen in your amygdalin is it has enzyme called as emulsin. Now this emulsin contains amygdalase, prunase and hydroxynitrilase. So let's understand them. So first thing, amygdalase will cleave up here. So this is removed and you just get a sugar out here. So now your amygdalin, once one sugar is removed, it gets converted to what is called as prunasin. Okay. This minus one sugar is prunasin. Now this prunasin is acted by the second enzyme called as prunase, and prunase will cut it out here. So your second sugar is also gone. You will be left with a hydrolyzed product of prunasin. Now this compound will undergo again a lysis in presence of hydroxynitrilase. Now this hydroxynitrilase, when this happens, will convert this hydroxy into a aldehyde. If it is a CH3 out here, you can call it as a ketone, but since it's a hydrogen out here, it will convert into an aldehyde, and so I'm just redrawing the whole thing here. I will get what is called as, this is my carbon, this is my aldehyde, and this is my H. So I get benzaldehyde plus hydrocyanic acid. So your amygdalin gets converted by amygdalase into prunasin. This prunasin is then acted about by the enzyme prunase, which converts it into hydroxynitrilase derivative, that is your benzaldehyde derivative. But this is further converted by your enzyme hydroxynitrilase into benzaldehyde and hydrocyanic acid. So your bitter almond oil, that is the oil which is obtained by expressing or squeezing out, is rich in benzaldehyde and hydrocyanic acid. On an average, your almond contains about 1 to 3% of amygdalin, and that's the reason it's very bitter in nature. This compound is not present in your sweet almonds. So you can check whether you're consuming sweet or a bitter almond by doing some tests which are particularly specific to amygdalin because of its ability to release hydrocyanic acid or HCN. Now one such test is ferriferrocyanide test. What you got to do is you have to extract, take a gram of drug, crush it. The moment you crush it, the enzymes as well as your amygdalin is going to come in contact and the process of lysis will begin. Now you can also facilitate the process by adding your alkalis. In this case, your alcoholic KOH is also facilitating the hydrolysis. Now once the hydrolysis is done, the drug is kind of boiled, complete hydrolysis is taken care of, you add a aqueous solution, a mixture of almost equal quantities of ferrous sulfate 2.5% with ferric chloride, and then again heat it for 10 minutes. You take this solution and transfer this test tube solution to another test tube which contains about 20% hydrochloric acid. You will see a nice Prussian blue color appearing, and that's due to the presence of hydrocyanic acid. Hydrocyanic acid is also known to precipitate mercury from your mercurous nitrate solution. So when you take your any almond, especially the bitter almond, crush it, heat it, slightly warm it, but you don't extensively heat it, otherwise your hydrocyanic acid will vanish. So if you are heating it, see to it that the vessel is covered, otherwise just very gently heat it and then treat it with your aqueous mercurous nitrate solution. You will see that after some time you will see some black color tiny particles or dots seen at the bottom of the test tube. That is nothing but your precipitated mercury which is being precipitated due to hydrocyanic acid. You can do also what is called as Grignard's reaction test. Grignard's reaction test can be done or can be observed again in most of the cyanogenetic glycosides which have the ability to release HCN. So what you can do here is you take your filter paper and you treat it with your sodium picrate. Now if you don't have your sodium picrate available, don't worry. You first dip it in picric acid solution followed by dipping it in sodium carbonate. So picric acid will form sodium salt and that's your sodium picrate. Now this sodium picrate filter paper, you allow it to dry for some time. So once it is dry, in a conical flask you will take your drug, maybe about 5 gram, crush it in mixer grinder with little water so that the enzyme and the cyanogenetic glycosides come in contact. As the reaction starts, HCN will go on releasing. Now as it is releasing, just hang this filter paper which you have soaked it in sodium picrate solution. Now this sodium picrate gets converted into sodium isoperpurate. Now sodium isoperpurate is brick red in color, but this reaction happens really slowly. So you will observe that after an hour's time, that filter paper which you have put inside the conical or kept hanging inside the conical gradually turns to a brick red coloration due to the Grignard's reagent. Another one is like with your guaiacum or guaiac resin. So you take your guaiacum or guaiac resin, dissolve the resin in alcohol. We want a filter paper, just slightly coated with the guaiac resin. So take this guaiac resin, ethanolic solution, dip your filter paper 2-3 times so that it nicely coats. A thin coat of guaiac resin is formed over it. Now this filter paper, you dip it in copper sulfate. It will acquire a little bluish coloration due to copper sulfate, and then this paper you keep in contact with a freshly crushed drug containing your cyanogenetic. You will give a distinct deep stain on the paper attributed to again presence of hydrocyanic acid. So this are few tests you can assure or you can run to check the almond samples you got, is it bitter almond, or to check if any drug contains cyanogenetic glycosides. Bitter almonds, like I said, are not safe for consumption. If you consume them accidentally, you will know them by the bitter taste. But still, I mean, on a smaller dose, people earlier used to give bitter almonds as a sedative. See, it kind of it's little toxic but causes sedation at low doses. The oil, still containing mild doses, is used as demulcent. Then the refined oil which is now free of benzaldehyde and free of hydrocyanic acid, is a good cheaper variant for your sweet almond oil and can be used in your skin creams and moisturizers as your emollient. Now this preparation, or this bitter almonds have also been used to prepare or purify amygdalin or even prepare bitter almond water for release of hydrocyanic acid. Now we go on to the next set of compounds, and that is the one we see a lot in Indian tradition, and the drug is mustard, and the category is what are called as isothiocyanate glycosides. So maybe you've seen people or those bodybuilders apply a little mustard oil on themselves. So sarso or mustard, you must have seen a lot of this cultivation of mustard happening across the northern India, especially Punjab, Haryana and the northern parts, plateaus, and also in the Kashmir region. So this consists of dried, matured seeds of Brassica nigra and Brassica juncea. These are cruciferae family members. This is the black mustard. If you are talking about white mustard, it is your Brassica alba. But white mustard is not as pungent or as spicy as black mustard, and as a result, traditionally, especially in Indian cuisine, you will find black mustard. Apart from that, black mustard has also been used for preparing sarson ka tel or black mustard oil. So geographically, it grows in the northern parts of India, China, Canada, and it's cultivated in North America also. So if you see this plant, it has nice beautiful yellow fluorescence, and then it bears these pods on maturing. These pods, if you open up, they will split, and within these pods will be your black color seeds. So these plants take about 90 days to mature. Once matured, they are nicely dried, they are threshed. During the threshing, the pods open up and release the seeds. They are then winnowed and the seeds are collected. If you cut open or break the seeds, you will see internally they are yellowish in color, and externally the seed coat or the testa is black in color. For Brassica alba, the seed coat is white in color. Now the black mustard contains compounds especially which are called as glucosinolates. Now, what are glucosinolates? You can see here there is a sugar, and glucosinolates are the category of what we call it as S-glycosides. So here the sugar linkage is attached to glycone by the sulfur. So glucosinolates are S-glycoside type of compounds. Now one glucosinolate derivative which is present in your black mustard is sinigrin. Now this sinigrin also contains nitrogen apart from sulfur, and there is an enzyme called as myrosinase. Myrosinase will specifically act on this. It will cut the molecule. Now because of its effect, when it is cutting the molecule, you will see that there will be some sort of rearrangement, and this rearrangement converts your this moiety into what is called as allyl isothiocyanate. So myrosinase is responsible for converting your sinigrin into allyl isothiocyanate. The typical aroma of sarso that you get is attributed to the allyl isothiocyanate. Now apart from it, it contains compounds such as caffeic acid, sinepin, and which are good as antioxidants and anti-inflammatory agents. If you press the seeds, the seed contain a very good or high content of oil, almost 30 to 48% fixed oil it contains, and of that oil 42% composition is a C22, okay, monounsaturated fatty acid called as erucic acid, and then you have about 12% of your C18 oleic acid. So you have your erucic acid and oleic acid, which are the principal components of sarso. Now when you see people rubbing it, in some cases the skin is very sensitive to erucic acid, and sometimes if you use the unrefined oil, the presence of sinigrin and allyl isothiocyanate irritates the skin. Now when it irritates the skin, it gives you the feeling of warmth, and this feeling of warmth increases the blood circulation, and that's the reason you'll see when your bodybuilders put this oil, you'll get a little reddish hue to the skin. So it acts as a rubefacient, improves your circulation, generates warmth, and that's how this mustard oil is beneficial. But if you want to consume it, it has to be refined. So during the refining stage, the allyl isothiocyanate, the sinigrin, as well as the enzyme gets denatured, and as a result, once this all are removed, it is much more safer for consumption. So applications, because it contains allyl isothiocyanate, this is there in most of the Cruciferaceae family members. It is known as a very strong antibacterial derivative. So it acts a very like a haptane and can inhibit bacterial growth. Because of allyl isothiocyanate and erucic acid, it acts as a counter irritant and rubefacient. Besides that, the oil extracted from black mustard is used for cooking, that's for culinary purposes. We now move on to the next set of glycosides, that is phenolic glycosides, and in this case we have a plant which is a beautiful orchid family member, and that's your vanilla. Now vanilla is a very exquisite, expensive spice or a flavorant which is used in food industry and is obtained from dried fruits of, or what we call it as the dried beans of, Vanilla pennifolia, or sometimes referred to as vanilla fragrans, which is your Orchidaceae family member. So being an orchid, it prefers a little moist, tropical, warm climate. It is not something which can bear frost, it's not something which can bear arid conditions. It prefers a little damp, moist and warm climate. So it's cultivated in mostly islands. If you go to see Madagascar, Seychelles, Mauritius, Sri Lanka, in the Indonesian islands, Reunion Islands. Also it's also cultivated in Mexico and some cases in the South Indian plateau region, where it matches the climatic conditions of your Indonesia and Sri Lanka. So some plantations have been actively taken in India as well, but majority of the supply of vanilla comes from the island nations. So this vanilla is actually a orchid plant. So cuttings are taken, they are grown, but you have to be very careful while sowing in the soil. Mostly it is allowed to rise vertically for a meter or two, and then it is allowed to kind of bend or turn on the plants. Now this plant, once it matures, like about 3 to 4 years, it starts bearing flowers, and this flowers just last for a period of a day. So now what has happened is, the important part to produce a fruit is pollination. The natural pollinator of vanilla is a stingless bee, which is now thought to be extinct, and it's not there in many of the countries. So as a result, people manually pollinate. So vanilla pollination is something which is mandatorily 100% done manually, you will see, and for that you have to take the style and stigma and kind of manually touch. So within that 24 hours, during the morning hours when the flower is most active and receptive, they say, that is the time you attach your style to stigma, allow little pollens to fall in. But this is not done for all the flowers, because if you do it for the whole inflorescence, it will lead to many fruit formation, and that will lead to a little smaller and not so good quality fruit. So selectively few flowers are fertilized, allowed to develop into fruit, and as you can see here, the beans are generated. Now this beans, if you go to see or if you take a whiff or consume, it will not have the flavor or aroma of vanilla. The reason being, in this particular pod, your vanilla is there as a glucoside. So you will have your vanillin glucoside, and this doesn't have the aroma. So you need to use the beta glycosidase. Now this beta glycosidase will act especially once the harvesting is done. So what is done is there are processing of vanilla beans. So once they are cut, they are kind of killed by putting it in a warm water about 60 to 65 degrees Celsius, by heating it and then removing it after 3 minutes. So that is what is called as killing, and then they are allowed to sweat. So they are kept in a closed box and let the moisture evaporate or come out of that beans. So during the sweating and during after that, this is followed by drying. So this is called as curing. So curing of vanilla bean is done, and just to show you, so these are your raw vanilla beans. During the curing process, you see that they develop color, they develop aroma profile because the sugar is hydrolyzed, and you get, this is called as your vanillin. Now this vanillin is the main aroma principle, and if you see good or nice cultivated varieties, they have from 2 to 2.5% of vanillin. Apart from vanillin, suppose if you change this to a COOH, you get what is called as vanillic acid. Suppose you change this to a CH2OH, you get your what is called as vanillin alcohol. And if suppose if you remove the methoxy, you get what is called as hydroxybenzaldehyde. Then you will get your hydroxybenzoic acid, and you will have your para-hydroxybenzyl alcohol. So these compounds are present in your cured. The process of curing may last somewhere between 3 to 6 months altogether, and during at the end of curing, you will get a product of this type, which is soft, pliable, and ideally should not break open. So during the application, the pod is open, you will see numerous seeds inside. The seeds are scraped with a knife, which contains almost little crystals of vanillin also at times, as well as the seeds, and that is what is used in your food industry. In terms of applications, you will be seeing that vanilla has been used as a flavoring agent. It is used in perfumes. You might get numerous vanilla perfumes, and also in pharmaceutical application as a sweet therapeutic agent and as a sweetening agent. So here's few more references for additional reading, and thank you everyone for your time.